everyone, welcome to my channel Pure Biology, and today I come with another video, that is the DNA repair. Okay, so what is DNA repair? DNA repair is that first of all we have to understand what is the DNA repair, and here the maintaining genetic stability requires not only an extremely accurate mechanism for replicating DNA, but also mechanism for repairing the many accidental lesions that occur continually in DNA. And most such spontaneous changes in DNA are temporary because they are immediately corrected by a set of processes that are collectively called the DNA repair. So first of all, we have to understand that DNA replication is the process by which new DNA forms from the parental DNA. And during the formation of the new DNA, there may be accidental different factors are there. These factors try to change the actual structure of the DNA, and that's why if these changes may occur in the base sequences or sometimes the nucleotide sequences. So, or you can say that the segment of DNA must be changed from the original DNA. So, in that situation, if there is any change occurring in the DNA segment or DNA base pairs, so during the replication, these are all corrected immediately. Okay, and this correction is done by different processes, and this is called actually the DNA repairing process. Okay, so this DNA repairing process is general type. Many types are there. Okay, generally first one is the direct repair, second one is the excision repair. Excision repair is there are two types excision in the excision repair. One is the base excision repair and another is the nucleotide excision repair. Third one is the mismatch repair. Fourth one is the recombinational repair. Fifth one is the repair of the double strand DNA break, and sixth one is the SOS response. Clear? So these are the different types that are related to the DNA repair. Sorry. Okay, so these are the actual types. So I will try to explain these different types of the repairing mechanism of DNA that are generally continually occurring in the replication process. Okay, or the you can say the DNA formation process. Now, the first one is that the direct repair. What is that direct repair? This direct repair actually these act on directly on the damaged nucleotides and converting each one back to its original structure. In this process, it directly acts on this. In this process, some mechanisms are there where the nucleotides are directly repaired to the original process. Okay, and here a few types of damaged nucleotides can be repaired directly. One very common types of UV radiation mediated damages. What kind of damages are repaired here? If any DNA nucleotides that are damaged by the UV radiation, so here the DNA may be changed or converted to its actual original position by this process. And here the pyrimidine dimers, you all know the two types of nucleotides, one is the purine, another is the pyrimidine. Okay, so I will try to explain about the purine and pyrimidine if I will give any lecture on the DNA structure. Okay, in that situation. So So there are just you just mind it that there are two nucleotide bases. One is the purine, another is the pyrimidine, that completely form the DNA double helical structure. So these pyrimidine dimers, these are damaged and these are repaired by the light dependent direct system, and by which process this is repaired, this is called the photoreactivation. Why this is called the photoreactivation? Here is the factor that is needed for the repairing process is that light. Okay, this is the light. That's why it is called the photoreactivation. And there is another important thing is that enzyme. Which type of enzyme is important for this mechanism? The enzyme is the photolipase. Okay, enzyme is the DNA photolipase. DNA photolipase. This is involved here. And when stimulated by light with a wavelength between 300 and 500 nanometer nm, then enzyme binds to pyrimidine dimers and converts them back to the original monomeric nucleotides. So what is happening here? The stimulation occurred by light. What wavelength is very important? That is 300, between the 300 to 500 nanometer nm. And then enzyme, when this is happening or the stimulation is happening for the DNA, then enzyme, that enzyme is the DNA photolipase. Immediately this enzyme binds with the pyrimidine dimers which already is damaged and converting them back to its original monomeric nucleotides. This is the direct repair. It's another example is also there, and I will explain after rubbing this whole thing. So this is the direct repairing process by which the pyrimidine base pairs or nucleotides, damaged pyrimidine nucleotides, these are totally repaired. Okay, and the enzyme is the DNA photolipase, and which wavelength is very important, that is the between 300 and 500 nm. Okay, so this is the direct repair, and is another example we will learn here just after rubbing this process. Okay, second example is that repair of O6 methyl guanine. This O6 methyl guanine that forms in the presence of alkylating agents, and this is a common and highly mutagenic lesion, and it tends, what's the actual function of this? Repair with the thymine rather than the cytosine. Repair with, it tends to pair with thymine rather than cytosine during the replication, and direct repair of O6 methyl guanine is carried out by the O6 methyl guanine DNA methyl transferase, which catalyzes the transfer of the methyl group of O6 methyl guanine to a specific cis residue in the same protein. Okay, so what is saying that? This is saying that here O6 methyl guanine forms. How this is formed? This is formed in the presence of alkylating agents. Okay, and this is a common and highly mutagenic lesion. And it tends to repair. What is the function of this? It tends to pair with thymine rather than cytosine. So actually in the DNA sequence, guanine that pair with cytosine and adenine pair with adenine that pair with thymine, okay, or thiamine you can say. So ATGC, this is the actual sequence of the DNA double helix. So, or you can say the DNA strand. But here the presence of alkylating agents, that is O6-methylguanine, the tendency of this guanine actually bind with thiamine or thymine rather than cytosine. Okay, and this is totally repaired by the presence of DNA methyl transferase enzyme, or the full form is O6-methylguanine DNA methyl transferase. This is the actual enzyme, and its main function is that it converts the methyl group from this guanine that is attached here. Then it just transfers the methyl group from the guanine and form the cysteine residue or cis residue, okay, in the same protein. This is occurring due to presence of the alkylating agents, and this is the example where the thymine dimer is formed. Why it is called dimer? You can see that two are attached with each other, not in the separate situation. That is called the dimer. Each one is called the monomer, and when two are bind with each other, that means it is called the dimer. So here the presence of UV rays, that dimer is formed, which is repaired to form the monomeric thymines or thiamines. Okay, it is called the thymines. So this is the DNA repair, or this is the direct repair, that is the one type of the DNA repairing system. And I will continue this repairing system or the next part in the next video. So you just continue with my channel. Okay, thank you.